Hello, everyone. I'm Kaori Sakata,
I'm Luz Almanza.
I'm Sarah Kim.
and today we're going to talk about the rabies virus.
During the period 1993 to 2002, 37,377 cases of rabies in dogs were reported, and 57,184 rabies cases in other animals were reported as well. The majority of them are in raccoons, skunks, bats, and foxes. Cats, through contact with rabid dogs or as predators of bats and other small wild mammals, play a role in transmission of rabies to man. Each year, nearly 1 million people seek care from the health system following an attack by an animal. Around 30% of them receive prophylactic treatment against rabies.
In the United States, most rabies cases are found mainly in wild animals, rarely dogs due to vaccination requirements. Only 1% of all reported rabid animals were dogs compared to the rest of the world, where it's over 90%. Approximately 55,000 people get post-exposure prophylaxis each year, which consists of a dose of human rabies immune globulin.
Rabies virus belongs to the Rhabdoviridae family and the genus Lyssavirus. It consists of negative sense single-stranded RNA, and it is an enveloped, bolus-shaped helical virus. This is a zoonotic virus, so it transmits from one animal to another. The mode of transmission is usually from saliva from an infected animal. Transmission has been observed in corneal and organ transplants, but this occurs where rabies virus infections are rare and can be misdiagnosed. In the US, there has only been 2 recorded cases since 2008. Sometimes it can also be transmitted through aerosol, but in order for this to happen, the concentration of the virus has to be very high. It's also a neurotropic virus, so it is both very neuroinvasive and neurovirulent.
The infection cycle begins in the muscle tissues that crosses neuromuscular junction to peripheral nerves and uses retrograde axonal transport to reach the central nervous system, where replication occurs in the muscle. There are multiple routes to reach the CNS via motor or sensory neurons, depending on the site of inoculation and post-infection time. The initial spread of rabies virus is retrograde, and at the end, it reverses in the direction of the transport. And if you look to the image on the slide, you can see that the retrograde starts at the axonal terminal and goes to the cell body, and anterograde starts from the cell body and goes to the axonal terminal. And the rabies virus uptake is mediated by clathrin-mediated endocytosis. There's also asymptomatic and clinical signs of neural dysfunctions that are presented in the later in stages. The infection deteriorates the health of the host that leads to death from the respiratory and heart failure.
In the rabies viral life cycle, the virus binds to the cell surface using glycoproteins and enters via endocytosis. Then its viral membrane fuses with the endosomal membrane to release the viral genome. The negative stranded RNA genome is transcribed by the polymerase complex, starting with a short uncapped leader RNA followed by the transcription of the 5 end-capped and polyadenylated mRNAs and their translation into the viral proteins.
The virus encodes only 5 proteins, which are all very important for its biological success. First, we have the nucleoprotein, which is responsible for encapsulating the RNA genome and protects it from degradation by RNases. The phosphoprotein, which is the non-catalytic subunit of the polymerase complex, the matrix or M protein, which makes up the membrane right underneath the envelope, the glycoprotein, which makes up these glycoprotein spikes localized on the envelope. These are able to bind mostly receptors present in neural tissues in mammals. Lastly, the polymerase protein. Since this is an RNA virus, it's unable to use host cell machinery for replication, therefore it needs its own RNA-dependent RNA polymerase.
Replication then occurs and the full-length antigenomic RNA is encapsulated in the nucleoprotein along with the genomic RNA. The synthesized antigenome functions as a template for the synthesis for multiple copies of genomic RNA. Lastly, the viral components are assembled and the rabies virus variants bud and are released, starting a new round of infection. The variants enter neurons by binding to a neural cell adhesion molecule. Following uptake by clathrin-mediated endocytosis, the rabies virus virions are transported within the vesicle and are released in the cell body of the infectious neurons, where replication and transcription occur.
Upon exposure to the virus from an infected animal through a bite, the rabies virus will multiply in the muscle tissue and then enter the peripheral nerves at neuromuscular junctions. It then spreads from the nerves to the central nervous system by retrograde axonal transport. The virus will multiply there in the central nervous system, causing encephalitis. It again will spread along the peripheral nerve to multiple body parts, including the salivary glands. Here it can be shed from the saliva. Susceptibility of infection of the virus can depend on various factors, including age, viral strain, dose, and distance between point of entry and the central nervous system. So the further away the bite is to the brain, the longer it will take for it to get to the CNS and the longer the incubation period. In rabies virus, the incubation period can vary a lot compared to other viruses and can range from 2 weeks to 18 months. The dose of rabies virus administered during exposure can influence the incubation period. Higher doses contribute to a more rapid deterioration of the host and a shorter incubation period. Lower doses lead to longer incubation period, and it can possibly increase the chances of an abortive infection. It is very important that post-exposure prophylaxis is administered during this time, since symptoms have not yet developed.
After the incubation period, symptoms will develop, and once they do, the disease is 100% fatal. Rabies virus affect the brain and spinal cord, so it causes neurological symptoms. Some symptoms include muscle spasms, fever, aggression, physical pain, mental confusion, hydrophobia, and paralysis. In the rabies virus, the skin is the first barrier. It evades the innate immunity both actively at a cellular level and via its tropism, which leads to the immunological privileged nervous system. A lack of an immune response contributes to the lack of symptoms during incubation period. The rabies virus inhibits interferon signaling, impeding an adaptive immune response until the viral load is high. The virus is highly immunogenic when given a vaccine, and in fact, when it is given immediately and appropriate to someone, it is almost 100% effective. There have been studies done that show that rabies virus cleared from the central nervous tissue by using immune mechanisms. In the experiment conducted by Roy and Craig Hopper, their goal was to clear the RV strain challenge from virus standard F3 from the central nervous system. This included the infiltration of immunological effectors across the blood-brain barrier, as well as the development of an innate immune response in central nervous tissues, adaptive rabies virus-specific immunity in the peripheral lymphoid organs. In the experiment, they used mice and infected them with silver hair bat rabies virus. They thought that the latter was linked to an enhanced blood-brain barrier permeability to the fluid phase markers, especially in the infected mice cerebellum. In method 1, they infected the mice with the silver hair strain bats rabies virus, which caused the BBB to stay intact, and the immune cells do not reach the central nervous tissue, causing the virus to not clear and causing the animal to die. In the second method, they enhanced the BBB permeability by inducing an autoimmune central nervous system inflammatory response, which increased the immune cell access into infected central nervous system tissues and aids viral clearance, averting the infection's deadly conclusion. And that wraps up our presentation. Thank you for watching.